Good morning once again. My name is Adeoti Olatunde Michael. This is a continuation of our discussion on non-Mendelian inheritance. Earlier, before in our earlier videos, I was able to look at the Mendelian principles. The Mendelian principle, where we mentioned three important laws of Mendel, according to Gregor Mendel. The first one was on the law of germinal unit, or what is called the law of monohybrid inheritance. After that, we also look at the law of dihybrid inheritance, which is also known as the law of independent assortment, and that is actually dealing with in, where we consider two traits or two characters at the same time. That is, the shape of any plant would not directly or indirectly, that is, it is going to be independent. The shape does not actually control or does not actually depend on whether the plant is going to be tall or whether the plant is going to be short. However, what we have is that the two traits or the two characters will be independently assorted. So we look at that and we were able to establish, according to Gregor Mendel, who actually used garden pin plants to verify most of his theories at that time. And don't forget that Mendel's postulates actually gained attention more than a century after he has even left. So, and that is why up to today, Mendel is still regarded as the father of genetics. So he postulated, he put across different types of like three or four laws, and what we actually know is that the traits of any plant that will obey Mendel's law must be that it must be dominant. One character must be dominant, whereas the other one will act as a recessive. And that was what we actually saw when we make use of Punnett square in verifying the law of Mendel. The first law was actually verified by the use of Punnett square, and whether it is first law or second law, what is very paramount is that the first filial generation of Mendelian inheritance or Mendelian plants must be such that it is going to be heterozygous. All those characters, the first filial generation is going to be heterozygous. And don't forget that we get the second filial generation by the time we cross the product of first filial generation. That means we are going to cross heterozygous plants. We will cross it together in order to be able to have what is actually known as gametogenesis. So that is very certain. The third law was the law of dominance, which I have actually said that Mendel believed that in his own plant, that is in garden pea plants, most of the laws, most of the genes were said to be dominant. One will be dominant and the second one is going to be recessive. So that was what Mendel used his lifetime to do. But don't forget that Mendel has just propounded all these theories, and all these theories must be verified by even scientists that will live after him. And that's the reason why that immediately after Gregor Mendel, most of his postulates were validated to be very correct. And the second thing many of the scientists that actually lived after Mendel did was to look at other plants, instead of focusing on garden pea plants, they look at other plants, and by the time they use other plants for this, they were able to now come up, they are able to now come at a point that not all plants will obey Mendel's law, but those ones that will not obey Mendel's law, they are regarded as the Mendelian principle of inheritance. That is why. And after that, scientists now came up and they discovered that non-Mendelian non-Mendelian inheritance pattern was as a result of more than about 10 new discoveries after Gregor Mendel's law. The first one is the law of incomplete dominance. It was after Gregor Mendel that they discover a phenomenon or a condition that is called incomplete dominance, whereby we have some genes that will be incompletely dominated. So instead of having one to be dominant and the other one recessive, so we now have one that is going to be incomplete dominant. We are going to see examples, and we are going to consider examples of this. What I'm just trying to do now is just this has been listed for us on the board. So the first condition is incomplete dominance. The second condition is codominance. There are certain genes that are said to be co-dominating. An example, we are going to see example of this when we consider different conditions, when we consider examples of all this. Then the third one is the law of multiple allele, and that is very, very possible in ABO blood grouping, that is in blood grouping in case of transfusion. So that is that. This fourth condition is some genes that are polygenetically inherited. So they are polygenically inherited, and we have seen a lot of examples on this as well, and we are going to see it as we proceed in this course. The example of this is what we have in human skin color. Human skin color, where we have each having contribution to overall phenotype, that is the skin color, is an example of polygenic inheritance. Okay, so the fifth condition is what is known as epistasis. There are some characters or traits in mice color that they are epistatic in nature, and we are going to see what epistatic actually means. So epistasis occurs when the expression of one gene is affected by one or more of the other genes. So that is a condition of epistatic, and this one is found in mice. The sixth one is genetic linkage or genetic recombination. Some genes are said to be genetically linked to each other. So that one will now make them to be able to produce a new product, which is going to be a genetic recombinant. We are going to see examples of this as we go on. The seventh condition is mitochondrial inheritance. Scientists after Gregor Mendel discovered that there are some that mitochondria, being a self-autonomous organelle in a cell, they have their own DNA as well. So that one now made them to behave semi-autonomously to the main chromosome or to the main gene of any plant. And we now discover that this one also deviated from the simple principle of genetic Mendelian inheritance, which has to do with dominance. So then number 8, which is very, very important in this course. Don't forget that this course is the title of this course is genetics and the environment. So we discover that their environmental play a very vital role in determining the genetic makeup of an individual of some certain plants. So we are going to see some of these as we go on. This 9th one is that there are some set of genes that are sex linked. So there are some genes are found in male, but they are not found in their female counterparts. So these are what is actually called sex-linked gene. And anticipation is just a phenomenon where a genetic disorder becomes more severe, and it appears at a very tender age. It appears at a very tender age of an organism, especially in their successive organism and in their successive generation. So these are part of what we are going to start considering one after the other. So what I will be doing is just that I will be picking them one after the other so that we will be able to know exactly what we mean. But we are going to start on how do the chromosome or the genes of plants that will obey Mendel's law, how would they look like, and what are going to be differences between the Mendelian chromosome and the non-Mendelian chromosome that will allow non-Mendelian chromosome to behave differently. We are going to see this as we actually go on, okay? So the first thing we are going to look at is the first one is I said we are going to look at how does Mendelian chromosome, how does it actually look like? Mendelian chromosomes look like this. Don't forget that chromosomes, they exist in pairs. They exist in 2, okay? So this is a pair of chromosomes. So let's assume that this is a point that is responsible for purple flower. Purple flower or white. So on the same locus, on the other hand of this chromosome will be, so this one, if this, okay, don't let us use purple, let's use red flower, okay, red flower. So the allele for that red flower, if this is capital R, the allele will be located exactly on the same spot in the second arm of this chromosome. Is going to be located and is going to be recessive. That is going to be recessive. That is a condition or that all the plants or all the organisms that will follow Mendel's law, that is how their own chromosome is going to look like. And throughout the life of a Mendel, he did not encounter a single situation that deviated from this. Don't forget that Mendel actually make use of garden pea plants, and that is the pattern of Mendel inheritance. So, and the first one So the first one, which is the first deviation, is going to be incomplete dominance. Okay, incomplete dominance is actually found in a special plants. So in a special plants, where we have okay, in a special plants, especially like a 6 o'clock plant, they discover that the color of the flower is either the color of that flower is either white or white, which is or it can be red. So if it is red, it is represented by, according to the observation of Mendel, according to what Mendel has actually put forward, so that if you have red flower, the opposite of it, which is going to be the recessive, is going to be white. So that means this one will be represented by capital letter R, whereas this one will be represented by R, which is small letter r. So going by this, that means if you now cross these plants, if they undergo the process of gametogenesis, okay, so this means that this is going to be capital letter R, small letter r. This is first filial generation. Don't forget what I said about Mendelian plants, that all the first filial generations are going to be heterozygous. So that means all of them will be red. All of them will be red. You can also use a Punnett square to verify this claim. But what is going to happen is that the first filial generation is going to be heterozygously red. But don't forget that there's no way we can actually differentiate, because the red color will occur or will appear at a phenotypic level, is physical. So everybody would think that it is red. We'll see the redness. However, when we now undergo, or if we want to look at the genotype, that's when we now see that it is heterozygous. Now, when you now want to get the second filial generation, don't forget you are going to cross the F1 together with F1. That means you are crossing capital letter R and small letter r, and capital letter R and small letter r. So that at the end of the day, we are going to have capital letter R, okay? This is small letter r, this is small letter r, this is capital letter R, and this one is small letter r. So under normal circumstance, according to the Mendelian principle, we will discover that the second filial generation should not even bring forth anything that should be heterozygous again. Should not at any point in time. Oh, sorry. Since it is a genotype, it's going to bring it. So that under normal circumstance, we were supposed to get, phenotypically, we are supposed to get 1:2:1. But scientists now discover that instead of them to get 1:2:1, they now got 3 different three different colors of this flower. So they got some in the ratio of 1 to be red. That means those one resemble the parents, their parents. It resemble one of the parents, which is very, very red. And the second one, instead of it to appear to be red. The second one now appear to be pink. This is red. This is pink. And the last one in ratio 1 appear to be white. So it is those ones that look exactly like their parents, they are red, and white that look in ratio 1 and 1. But those ones that now have pink color, they are now in ratio 2. So the question is, where is this third color coming from? So that was why they now discovered that this one did not obey Mendel's law. That is, it has deviated from Mendel's law, and this is very common in some plants. And that is exactly what they call incomplete dominance. You will see that had it been this capital letter R for redness is completely dominating with those supposed to have the third color, and that's why it is now regarded as incomplete dominance. So if anybody asks you to give or to write or explain what are those plants that will not or does not obey Mendel's law, so the first phenomenon is on incomplete dominance, and that is what happens in these plants. I'm going to give you the example of plants that will undergo the law of incomplete dominance. This is very, very common in a plant which is called snapdragon. It's called snapdragon. Snapdragon, very common there. So they discovered that this, and the botanical name for that plant is called Anirinium majus. So this is an example that obeys that did not conform with the Mendelian principle, and the reason being that the first generation, the parental generation, is red. So they now cross the red with white flower. They discovered that the first filial generation all were phenotypically red. But when they now cross the product of all that were phenotypically red, when they now cross them together, instead of them to still obtain red and white, they now obtain the third color, which is called pink. And the question is, where is that pink? Where is it actually coming from? So, and the assumption was that normally, that instead, look at this chromosome, Mendelian chromosome, where we have red and we have white directly located on the same locus with the red. So they now discovered that there are some genes that are responsible for the color of the flower, that they might not be located on the same locus. So if these genes are not located directly on the same locus, that means they are not going to obey the law of dominance and recessiveness, and that is exactly what leads to what is called incomplete dominance. So, and we have given you example of that. So the second example we are going to consider is that of codominance, the law of codominance. The law of codominance. Don't forget that it is better you draw something like this to actually illustrate what is actually expected in the law of Mendel's. Okay, so codominance. So this codominance, so the second one is a dominance. So they now discover that there are some genes, that have the same, they have the same, for example, in the ABO block grouping. So A has the same chance as to be dominated, I mean to dominate as B. So A and B are co-dominants. Again, under normal circumstances, we are talking of Mendelian principle, which is a Mendelian inheritance. One of these two characters, one will be dominating, whereas the other one will be recessive. But in the case of co-dominance, from the word co, they are colleagues, they are together, they are like friends, they have the same opportunity. So in ABO block grouping, A has the same chance to dominate as B. So that is the example of where, and this is very possible in human blood grouping. You know that human blood grouping, you know that, okay, let's say this is the red blood cell. Let's say this is the red blood cell. So if this is group A, this is group B, this is AB, and this is group O. So the antigen, so let's say on A now, so if A is like this is A, and B is like this, which is rectangular, that means anyone that will be A will contain something like this. Anyone that will be B will contain something like this. Anyone that will be AB will have the two, going to have this as well as having this. And anyone that has neither A nor B, so it's going to be like this. Okay, only the presence of A does not affect the presence of B. And that is why it can come together, and by the time it comes together, it does not exercise dominance, and none of the two alleles will exercise complete dominance over another one. So that is what is called the law of codominance, and it was found out that the ABO blood grouping in human blood grouping does not obey the law of dominance, because the presence of A and B, they have the same equal chance. The presence of one does not affect the second one. No one is acting as recessive, and no one is actually dominating. So that is why they call them colleagues, co-dominant. So we are going to see the principle of this ABO blood grouping as we go on. But you should just know that ABO is being grouped according to the presence or absence of B. The presence of, or the presence of A or B, A and B, or neither. So if this one is A, that means it has A antigen, which is like this, which is triangular. If it is B, it has antigen B, which is rectangular. And if it is AB, it has both rectangular antigen as well as triangular antigen, and that's the reason why it is AB. But this one that has neither A nor B antigen, nothing. This one cannot actually react, doesn't have anything to actually react with. And that's why if you give any of these groups, if you give them their whole blood, that's why they regard them as a universal donor. And AB, which has antigen for A or for B, if you give them A, they will accept. If you give them B, they will accept. If you give them O, they will accept. And if you give them AB, they will also accept. So that's why AB is regarded as a universal recipient. So they now find out that there are some genetic factors that actually determines the presence or absence of either A antigen or B antigen. So, and all these factors is what we must know, in which is applicable in medicine today, especially in blood transfusion. So that is the second condition that does not conform to the principle of Mendelian inheritance, which is the dominance, law of dominance. Okay. The third one is the law of multiple allele. Multiple allele. That's the third one. Multiple allele, or what you call so it's still the same thing. It is still the same thing as codominance. Blood grouping in human. Also, blood grouping in human, which is ABO, actually fulfill the law of codominance as well as multiple allele. Multiple allele. Because the other one, this one is going to be is going to have antibody for B. This one is going to have antibody for A. This one is going to have antibody for AB, and this one is going to have no antibody. So that it's going to be opposite. If we are in microbiology, that is medical microbiology or medicine, we'll have actually gone ahead in immunology. I've actually gone ahead to know the implication. What do we mean by antigen? What do we mean by antibody? What is antigen-antibody reactions? And what are the likely? What are the likely? And that's the reason why in some places, let's just be silent about it. So I said number one is incomplete, number two is codominant, number three is now what? Is now multiple allele. And you find out that ABO blood grouping is also an example of a deviant gene, of deviant genes from the Mendel's law, deviant genes from Mendel's law. Okay. So this fourth one is polygenic inheritance. The law of polygenic inheritance, which is number 4. Polygenic inheritance. This one is actually found in skin color. In skin color. So they now discover that okay, let me try and see, make use of some illustration of polygenic. Okay, now please make use of this diagram so that you'll be able to understand what is called non-Mendelian. The fourth one we said incomplete. Abi? So if this one is allele 1, this one is allele 2. This one is allele 2. Their point of intersection here, the allele 1 is red, the allele 2 is white. So the point of their intersection here is pink. So where is this one actually coming from? That is incomplete dominance. That's a place whereby you have set A and B, allele A and B, with point of intersection of a different color. So that is an incomplete dominance. Now, codominance factor. Now, codominance factor is when you have okay. Now, when you have allele 1 and this, none of this one act as dominant, and none of this one is acting as a recessive. None. This one is not dominating, this one is not dominating. You will discover that at the end of the day, when we look at this, it was even pink that was, because what we got was 1, 2, 1. So this one is not dominating, this one is not dominating. It is even the third color that is dominating. That is a condition for incomplete dominance. Okay, now codominance this time around is when you now have the same diagram, and you consider the same length. You have to be careful that there is a space between here, and in all these three, they are the same, so that it's going to, so this is codominance. So this is allele 1, this is allele 2. This is talking about allele A, which is antigen A, and this is talking about antigen B. So if the third condition is neither, or both, which is you have both. So this one is going to be O, this one is going to be AB. So that is an example, a typical graphical illustration of what is codominance, as well as what you can also term as being so that is incomplete, that is codominant. Now the third one, which is now the law of multiple allele. This time around, you have three allele. The first one is represented by black, the second one is represented by blue, and the third one is represented by red. So if this is allele 1, this is going to be allele 2, and this is going to be allele 3. So this is like this is A, B, and O. So this one is what is called multiple allele. You have more than 2. In incomplete, you have 1. You have 1, that is the difference. So, and the second condition, we have 1 that is dominating. The second one is that the allele, neither the allele that is found in either of the parents, none of them is now acting as a dominant gene or dominant allele or even recessive. So there's nothing like a recessive or dominant. Now in the second one, which is the law of codominance, both of them, both allele 1 and allele 2, they have the same tendency, and that's why they call them what this. But in the case of multiple allele, that is when you have more than 3 allele now. So you just have that there will be a point here of intersection of 2 and 3. There will be a point here of intersection of the 3. Then there will be a point here of intersection of A, that is 1 and 2. then there will be a point here whereby you have A and B. That is exactly the simplest way you can illustrate the law of the law of multiple allele. The law of multiple allele. So that is that. Now, when you now have polygenic traits, that is when more than one gene is controlling a single trait. So let's say you now have that's why it is polygenic. You have 1, 2, 3, 4, 5, but you have more than one gene that is controlling a trait. 1, 2, 3, 4, 5, 6, like that. So this is a single trait. And I told you that this one is found in skin color. This is found in skin color. Is a kind of a non-inheritance pattern that is found in human. It's also found in human. So the fourth condition, sorry, the fifth condition now is genetic recombination, or what is called genetic linkage. Genetic recombination, or what is called genetic linkage. They just discovered that at certain points in time, okay, at certain points in time, especially in during, okay, number five now. So number 5, this is where you have genetic recombination, or what we call genetic linkage. Genetic recombination, or what you call linkage. So anytime that you come across, like, if you are given a kind of some genetic questions to solve, and at the end of the day you now get equal, like 1:1:1, or 4:4:4, or you get 2, so, or you get the same whenever you have something like this, 1:1:1:1, or 4:4. This one is telling you that that gene of interest, that gene in that particular organism is what is genetically linked. And that's what is called recombination. And I told you that this one is found in meiosis, especially during meiotic division, whereby you have a formation of okay, let's make an illustration. Now let's say you have 2 chromosomes. If you have 2 chromosomes, like this, so at this point where the harm of this one is causing the other one, there's going to be exchange of genetic materials, between these 2 arms of chromosome. So that at the end of the day, during the process of meiotic division, especially during formation, this is exactly what is called formation of chiasmata. That is at meiotic division. So formation of chiasmata. When this one has exchanged its genetic material, at a point in time, there will be segregation. There will be segregation whereby all the chromosomes will now align at equator. This one will form at the equator at northern pole as well as southern pole, where there is going to be pulling together of this. So there is going to be genetic exchange of materials. So whenever they now give birth, part of, there are some chromosomes that must have contained combination of both father and mother genetic information, and at the end of the day, if those chromosomes now go into meiosis or gametogenesis, they have now discovered that they are not going to follow the Mendelian pattern of inheritance, and that is the fifth condition of definition from Mendel's law. So that is called genetic recombination, or what is called genetic linkage. Genetic recombination or genetic linkage. Don't forget what we have done here today. We have been able to re-examine again, repicture the chromosome, Mendelian chromosome, whereby you have a gene or an allele, a gene here, and the allele is going to be located directly at the same locus with this, so that one will be acting as dominant and the other one will be recessive. That is what happened in garden P. But immediately after Mendel's law, after Mendel must have gone, I told you, more than almost a century after what he has done, after his work was published, that was when scientists after him started to rework and revalidate what he has said so far. And they now discovered that there were 10 different cases. Nowadays, there are 10 different cases that will not conform with the Mendelian principle of dominance and recessive. And we'll actually look at the first one, which has to do with incomplete dominance. The second one is codominance. The third one is multiple allele. The fourth one is polygenic inheritance. And the fifth one is genetic recombination. I have actually mentioned example of where that is very, very possible. Polygenetic in the sense that, oh, sorry, I told you that polygenetic inheritance is when you have one gene, many genes or allele that is controlling only one trait. So the gene that is controlling the skin color, is not just one gene, it's a multiple gene, polygenic gene. So, and that's why skin color does not actually and will not actually follow the non-Mendelian principle. The parents, okay, don't let's go into human genetics anyway. So after that is what is called genetic recombination. So I told you that during meiotic division, chromosomes tends to come together. By the time they align at the point of equator, if they align in the middle, the point of equator, that is at metaphase. Metaphase 1 in particular, at metaphase 1, whereby there is formation of chiasmata. So the two genes, the two chromosomes come together, and there will be, by the time they come together, before they will separate, there must have been exchange of some genetic materials, so that later on, when each chromosome is expected to move back, 23 pairs, you have 23 up and 23 down. So that 23 must have exchanged some of this genetic material together, and by the time they want to actually undergo the process of gametogenesis, they've just discovered that they will not exactly behave like the original unmixed chromosome of the parents. So these are conditions that will obey non-Mendelian principle, and it is at that point today that I want to conclude here. Don't forget that most of these there will be a time whereby I'm going to give you tutorials on all these tutorials and assessments of all this, and I'm going to tell you how you are going to do it during the coming time. So when we now come next time on genetics, we'll now consider the next 5 of deviation from Mendel's law. Thank you.